Fluorescence in situ hybridization is a technique used to visualize specific locations on a chromosome through a fluorescent complementary probe. This video will first give you an insight into the molecular principles of FISH, after which we describe the general lab procedure with which it's performed, as well as an example of its use in research. Throughout the video, we will highlight important points in the methodology of the experiment. To begin with, FISH aims to detect chromosomal abnormalities that could cause disease. By visualizing genetic sequences, it can tell us whether or not a chromosomal location is mutated. Thus, it can also be used for gene mapping or quantification of nucleic acid sequences of interest. In our case, FISH was used to determine the number of HPV copies in two types of cancer cell lines, Kaski and Sia, in order to analyze the physical status of HPV. The first step of the FISH assay consists of fixing cells onto the slide, causing protein crosslinks. After this, the probes are added to the DNA. The probe must be complementary to the region of interest on the chromosome. The probe can be directly or indirectly labeled. In our experiment, indirect labeling was used. To produce the probes, antibodies are attached to double-stranded probe DNA. This is then denatured and hybridized with the target DNA sequence on the chromosome. Now, an extra step is required for visualization of the non-fluorescent antibody via an immunological detection system. Though FISH is faster with directly labeled probes, indirect labeling offers the advantage of signal amplification by using several layers of antibodies, and it might therefore produce a signal that is brighter compared with background levels. The pretreatment of cells is essential for effective FISH. In this step, proteolytic digestion helps permeabilize the cell's membranes, which makes it easier for the fluorescence-labeled probes to penetrate them. Our experiment and tutorial begins by pipetting the cells onto the pre-coated slides and drying them on the heating plate. These are the slides that will be analyzed under the microscope at the end of the experiment. Drying is followed by the incubation of the cells in the pepsin HCl solution for 10 minutes. The pepsin used in this step serves to digest proteins attached to the DNA, making it accessible for the probe later on. It's important to use the correct pepsin concentration. If the treatment is too high, the sample will become fully digested and there will be nothing left to label. Finally, the cells are dehydrated in ethanol so the label can be added more easily. The labeling and probe preparation step makes use of nick translation, a technique in which DNA polymerase I replaces nucleotides of a DNA sequence with their labeled analogs. This creates a tagged DNA sequence, which can be used as a probe for FISH. Labeling assay is prepared according to the supplier's instructions. The components are first pipetted into tubes, then vortexed, and then centrifuged. The tubes are placed into a thermomixer which generates the necessary DNA fragments, whose length can later be controlled by gel electrophoresis. The denaturation and hybridization step serves to denature chromosomal DNA and hybridize it with the probe DNA. The mixture is prepared according to the protocol's instructions and applied onto the cells on each slide, after which they are covered with a cover slip. The denaturing takes place on the heating plate, while the hybridization happens in a humidified chamber in a hot air oven, preferably at 37 degrees Celsius overnight. After the incubation, the excess probe is washed off. When removing the cover slip from the slide, dip the slide into the buffer and then remove the cover slip carefully with tweezers. Done improperly, this can result in cell damage. In this experiment, a low stringency wash was followed by a high stringency wash. High stringency, done at high temperature and in a low salt buffer, permits only hybridization between nucleic acid sequences that are highly similar. After the wash, a blocking buffer is added to minimize non-specific antibody binding and noise. Antibodies for indirect labeling are added to the target DNA via the probe, after which the slide is incubated and washed. As we have several layers of antibodies, this step is repeated. Finally, we dehydrate the slides in order to remove the water so DAPI, a general non-specific dye used to stain the nucleus, can stain well. With fluorescent microscopy, these images are produced. FISH signals are visible but need to be analyzed further. The software ImageJ is used for analysis. The first step is to split the colors. Then, the threshold is adjusted to distinguish FISH signal from background noise. Using the Region of Interest Manager tool, select 5 regions that represent the background and do not contain any cells. The software will output the signal intensity as raw integrated density. Calculate the average raw integrated density across the 5 background regions. Subtract this value from the signal to correct for background noise. Now we are ready to quantify the FISH signal. Select the FISH signal of a single cell and go to Analyze under the Particle Analyzer tab. Select the parameters that you see on the screen. This tool will measure the signal intensity across the selected areas. Save the output and repeat for the other cells in the sample. The same analysis was done for the CS cell line on the right. According to literature, this cell line contains 2 HPV copies. We found that the signal intensity in the SIA is 470 for 2 copies. Dividing the signal intensity of a complete nucleus for the Quesky cell line on the left, we can estimate that these nuclei contain roughly 135 HPV copies. The image analysis concludes our tutorial. To summarize, the following points were covered. We illustrated the general steps involved in executing a FISH protocol. The context and purpose of some of the most important steps were explained. We discussed a few considerations with regards to producing reliable results. And finally, an example of the application of FISH in research was given. For more details on the steps and the full protocol used in this video, please scan the QR code.